All right. Okay, everyone, switch on your videos. Let's start today's class. Also, so in the previous class, we completed the human health and disease chapter. Did you people have any doubts in that? Hmm? No doubts? Marwa, please switch on your video. Only two of you have joined yet. Where are the other students? All right, so I was thinking today we will begin with a new chapter. Are you people able to hear me fine?
Yes, ma'am.
All right. So today we'll be starting with biotechnology, principle and processes. Okay, the first part of the biotechnology chapter. Now see, in this particular chapter, you're going to see how we are going to, integrate technology into the biological world and thereby create new products for, better human consumption or human use. So that's what we're going to see. Okay, so have you people like heard about biotechnology before? Have you or have you not? Also, Marwa, are you able to hear me? Please switch on your video. Also, when I'm done teaching the introductory part, like when I'm done teaching the part that I'm supposed to for biotechnology, then I'll make you people solve questions from human health. And are you people studying the previous chapters that have been done, that have been completed? Have you started to learn the chapters or the lessons? Yes or no? What is the smile? I can't make out what the smile is. Does the smile say no, ma'am? Yes, Marwa, speak. You can speak. It's okay. I will not eat you. I'm very far. You people haven't started to study, na? Your faces tell me that only. So start it. Even if you haven't, start it because it's very, very important now at this point because all of you are going to have your this thing midterms now, right? Half yearly examinations and stuff. So start the studying process. One by one, the chapters are also being done, right? Now tell me one thing. In the previous class, did we complete that chapter, human health and diseases? It was completed. Okay. So is there any part of that chapter that you people have a doubt in? No doubts, na? Okay. All right. So talking about biotechnology principle and processes. So basically in this chapter, you're going to see how the technology is being used in several fields such as agriculture, such as food science and medicine to be the most important one. So now the European Federation of Biotechnology, that is abbreviated as EFB. Now this says, like it has given the proper definition of biotechnology, wherein it tells us that what it actually is. So when we talk about the particular definition of biotechnology, what do they say? That the integration of natural science and organisms, the cells and parts thereof, and molecular analogs for products and services. Now, see, students, we have studied about the Human Genome Project and everything related to that, right, in molecular basis. Now, when we have studied about that, we have come to know how, like, when you know the genome of any organism, it becomes very easy to solve most of the problems that come associated, right? Like what all problems? Problems such as if there is any problem in the healthcare part, if there's any problem in the, you can say this part, what do you say, or agriculture part, right? So all of that can be, you can say properly done because of the genome sequence that we know of. So this is what they say, that we integrated somehow the alteration of the gene, alteration of the DNA is done in order to, you can say, make products that are worthwhile. Now, talking about the principles of biotechnology. So biotechnology is based on two principles. The first principle is genetic engineering, and the second principle is the bioprocess engineering. Okay, now see, genetic engineering, basically the whole entire thing, when you alter the genetic material. Now, alteration could mean subtracting something. Alteration could mean addition of something, right? Alteration could be both, subtraction, addition, both. So when you alter the chemistry of the genetic material and basically you changed it and then you put it into the host organism, and now you want to see how the host is reacting to that change. That is the genetic engineering. Second is the bioprocess engineering. Now this is when, while you're doing all of this stuff, obviously you're working with life, isn't it? So when you're working with life, naturally you have to maintain a sterile condition. What is a sterile condition? Sterile condition is the one which is contamination free, right? Free from any of the contamination. No germs, no fungus, no contamination from the outside must come. Then only whatever you're trying to grow would grow flawless. Is it clear, students? Any doubts? All right. And now see, when you are altering the chemistry of the genetic material, you obviously do it in a lab. You can't do it in your kitchen, isn't it? You have to need a proper apparatus and instrument for doing that. So when we talk about the things that we do in the laboratory, so what is the end product or what is the result that we get from it? So basically, the recombinant DNA is what we are after. So what is recombinant DNA? These are the DNA molecules we form in the laboratory. We do it. Humans do it. Imagine. And then basically what you do, for example, if there is, let's just say, there is plant A. Okay. It has a character that it can grow in a very high soil condition. Okay. This is A. And then there is plant B. Now it has a character that it can grow in all seasons. Okay, it can grow in all seasons. This plant can grow in high salt concentration but cannot grow in all seasons. This plant can grow all seasons but cannot grow in high salt. So what they did is that they take the, you can say, all season gene from this plant, the salt stress tolerating gene from the plant A, and then they combine it and put it in a plant B, I mean C. So now this plant is going to have the superior characteristics from A and from B equal. Is it clear? So the gene that you make on your own, that is your recombinant DNA. It is also written as small r DNA. So let us see what we have written. So these are the DNA molecules that are formed in the laboratory by using the methods of genetic recombination, such as cloning, that brings together genetic material from multiple sources, right? Just like how from A, from B, you took out the genetic material, incorporated into the genetic material of the C. And since all of you have studied about genetic material, nucleic acid, everyone is familiar with that? Are you okay? I'll say a few words. Tell me if you're familiar with that or not. Okay. First, DNA double helix. Familiar with that? Very good.
Yes, ma'am.
Diester bond. Familiar with that?
Yes, ma'am. Yes, ma'am.
Okay. Aisha, switch on your video. Also, okay, phosphodiester bond is done. Then polynucleotide chain. Familiar with that?
Yes, ma'am.
Okay, wonderful. Then what about ATGC? Familiar with that?
Yes, ma'am.
A pairs up with
Yes, ma'am.
A pairs up with G, right? A pairs up with G and C pairs up with P. Am I right or am I wrong? Right or wrong?
C pairs up with G, no?
So I am wrong, right? A pairs up with T and G pairs up with C. But A pairs up with T with 3 hydrogen bonds, right or wrong?
Sorry, ma'am.
A pairs up with T with the help of 3 hydrogen bonds, right or wrong?
Wrong.
What is the right thing then?
It's a
You're calling
double
your
bond.
ma'am wrong.
It's
You're
double
calling me
bond.
wrong. How could you do that? But yes, you people are right. It is A pairs up with T with how many hydrogen bonds? 2 hydrogen, right? Okay, very good. So this is the basic introduction of this chapter. Now you're going to see with the help of biotechnology how you are going to incorporate it in the field of medicine and create a personalized medicine for humans. How you're going to incorporate it into the field of agriculture and make better agricultural practices. And then how you're going to apply it to the field of variety of products that the humans can make. And then how could that help us later on? Understood? But in this particular chapter, you're not going to see the application because that's the next chapter. In this chapter, you're going to see how we are doing it, like the whole entire setup, how we are taking the DNA out, how we are cutting the DNA, how we are molding the DNA, all of that. Okay. So first, you people write down this much, all the points that are written this much, like this side, all the points that I've read, you write down that. Okay. The left side point, you don't write. Okay. So yeah, now you people note it down. Who has done this on the screen? Who did this on the screen, students? This is not something done from my side. Who did it? Tell me. Students, are you able to hear me?
Yes, ma'am.
Who did this? Who is drawing circles and stuff? Allah. Okay, see students, this is for me to write, okay, not for you. So do not write on the screen, okay? You just write in your notebooks. Done, students? Shall I scroll down? Students, are you done? Yes or no? Shall I scroll down?
Yes, ma'am.
Done, students? Shall I scroll down?
Yes, ma'am.
Done, students? Done till here? So this was about the introductory part of the biotechnology and its processes. Okay, now the next thing, we're going to move forward and see how the whole entire cloning process is done. So basically, like I told you, that cloning is the technique wherein you take a foreign DNA and then you incorporate it into the host. So if you see over here, the steps to do cloning. And what is the meaning of cloning? Cloning basically means making multiple copies that are identical. So when you make multiple copies of something, and that too identical multiple copies, we call that as cloning process. Now let us see the steps. First of all, here in this picture, you can see the DNA, which is the choice of your DNA. Okay, the gene of interest you've taken, you've cut it, and now you're ligating it, meaning you're joining it to a vector, which is basically a carrier. Then the end result is the recombinant DNA. Now, for the second step, this recombinant DNA is transferred into the host cell. For example, over here, I've drawn a bacterium, which is the, you can say the host cell over here. And then the multiple copies of that recombinant DNA is made. How? When that host cell will divide, so will the multiple copies. Understood? So this is how cloning happens. We take over the machinery of the host and thereby create the products that are of interest to us. So the first recombinant DNA, it was made by Stanley Cohen and Herbert Boyer in the year 1972. Okay. And then basically what they did, the whole entire process, they linked a gene of their interest that had antibiotic resistance. And then they linked it with the plasmid, which is, you people have studied about plasmids, like other than the genomic DNA, the extra chromosomal circular DNA in the case of bacteria, mostly the prokaryotes, that is a plasmid. So they took out the plasmid of Salmonella typhi, which causes typhoid, right? And then they made the whole entire thing. Now, how did they do it? The three basic steps that you utilize in genetically modifying an organism. So first, you see which is the gene of the interest. So identify the gene of interest, then introduce that gene into the host, and then maintaining that so that whenever the host will divide, the gene of interest will also divide and be incorporated in its generation. So these are the three basic steps. Is it clear, students? Did you understand this much?
Yes,
Yes
ma'am.
or no? So this is all in all the whole entire gist of the chapter. Now basically we're going to see what are all the tools that we utilize to make the recombinant DNA technology. Okay, what are the various things that are needed, how the process happens, all of that. But up until now, you people have understood everything? Yes or no? You've understood?
Yes, ma'am.
Oh, all right, now you note down. Then we'll talk about the tools that are required during the process of DNA, I mean recombinant DNA technology. Done, students? Shall I scroll down? Done till here? Is everyone
Yes,
done?
ma'am. Yes, ma'am.
Done, students? Shall I scroll down? Are all of you done?
Yes, ma'am.
Okay. Done, students? Done till here? Is everyone done? Yes, is everyone done till here?